Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here with a guest that I've been wanting to speak with for a really long time. He's also come very highly recommended from a few other people, and that's Pradeep Natarajan, a preventive cardiologist and cardiovascular geneticist at Mass General Hospital, where he's director of Preventive Cardiology, and honestly has been behind some of the most impactful work in early prediction of cardiovascular disease in the field over the last decade. So thank you so much for joining me. I'm excited for the conversation.
Thanks, Patrick. Thanks for the invitation. I'm super excited as well.
So the two things that I think about most when I think about the impact you've had on the field is clonal hematopoiesis and how we understand that risk in cardiovascular disease, and also polygenic risk. So those are the two areas that I especially want to dive into. We'll take this a lot of directions because you've also done amazing work in genetics driven drug discovery, in understanding LPA and other factors. So we're going to cover as much ground as we can in the time, but I kind of want to start with the clonal hematopoiesis story. The way I was reading your work as I was working through my PhD, and the way I remember it was probably about 10 years ago, 2016, 2017, that this seminal paper that you were leading came out and really redefined the field. I'd love if you could tell people a little bit of the backstory. Like, how did you get thinking about this, and what is it for people who aren't familiar with this really important branch of predictive cardiology?
Yeah, I mean, so I was also in training. This was also around the time, I was wrapping up my postdoc. And so the genesis of this work was happening earlier, deeper into my postdoc. But even walking before that, so in 2014, there were three papers that kind of, I would say, started the field related to clonal hematopoiesis as a population level risk factor. And this is because, largely at the Broad and many other places, there is more and more next generation sequencing that was being generated, specifically whole exome sequencing. And most of these were to look at the inherited basis of a wide variety of non-oncologic conditions, but also oncologic conditions. We were engaged in exercise for what are the causes of early onset myocardial infarction, David Altshuler's lab on type 2 diabetes, Steve McCarroll on schizophrenia. And kind of all of these groups and others kind of came to the observation that if you re-looked at the exomes that were being generated from the blood, they were coming from white blood cells because red blood cells are anucleate. Beyond the germline genetic variants that we were looking for, the loss of function mutations, the PCSK9 equivalents, that you could also identify somatic mutations. And a surprising observation for basically three papers in 2014 were that there was a high prevalence of cancer-causing mutations that were detected at the technical sensitivity of a whole exome. What that means is, the average coverage of an exome is about 90x. So for every base pair, you are reading that base 90 times. And so if you detect something, and for germline genetic variant calling, you're basically detecting genetic variants that are the exact same as the reference would be wild type, 50% different would be heterozygous, 100% difference would be homozygous. Somatic mutations would be somewhere, across those. And most of the time when you determine that, that's artifact, that's junk for various different reasons. But, there are tools when you're doing somatic variant calling. In general, for an individual, you have the tumor tissue and you have normal tissue that's often the blood. And you compare the two, and then you can say, okay, we have the equivalent issues related to artifacts, and we can kind of screen those out. But in the tumor tissue, there are these things that we're seeing that are not in the blood. Here, this is all coming from the blood. There's no additional comparator. but we can use population level exome sequencing data. Again, that was also being generated at the Broad to find mutations that were currently observed that were somatic. And we've specifically honed in on well-established cancer-causing genes and cancer-causing mutations. And just going back, them being detected at the technical sensitivity of an exome means that this is not happening in a one-off cell. This has to have happened in multiple cells. And if it's in multiple cells and it's a driver mutation, there has been proliferation or clonal hematopoiesis. And hematopoiesis itself is a polyclonal event. But with the driver mutation, it suggests that that driver mutation might be driving that mosaicism, that clonality. Okay, so the surprise in 2014 was that actually a lot of people had this.
Right.
And, blood cancer, the ones that are these mutations that are observed in blood cancer, is pretty rare in the population. It's like, 0.1% in the population. Okay. But again, as people age, the prevalence of this condition increases. But individuals in their 70s, about 10% of them had clonal hematopoiesis, had cancer-causing mutations that had expanded, and they had no cancer. They didn't even have dysplasia, and the blood counts were normal. That was a surprise. Obviously, there's an increased risk for blood cancer by about 10 to 11-fold. And there was an overall signal for mortality by about 40%. And this is observed in all of these papers. Now, so you might say, because it's a risk factor for cancer, that they're all like dying of cancer. However, as I said, blood cancer, the rate of blood cancer is really rare. So even a 10 to 11-fold relative increased risk, maybe that'll go from like 0.1% to 1%. So will not explain why all these individuals have a higher risk of death. And so in one of the studies that we'd collaborated with, especially using, data sets where there are some cardiovascular outcomes, there were cause-specific analyses that suggested that maybe it was the leading cause of death, cardiovascular disease, a mix of coronary artery disease and stroke. And so in one of the 2014 papers. And so that paper that did that is led by Sid Jaiswal, who was a postdoc at that time at Dana-Farber with Ben Ebert. Also, like all of us, I was at MGH, but we all kind of converge at the Broad. But I had zero reason to interface really with, Ben and Sid. But they were very good collaborators in kind of ascertaining these exomes. But around this time, 2014-ish, Sid and I had an opportunity to present to the Broad Scientific Council. And these are, leading geneticists across the country. Obviously, Eric Lander was there, but Joe Goldstein, who'd won a Nobel Prize related to familial hypercholesterolemia, was the chair. But you have other folks like Rick Lifton, Huda Zogby, and many other luminaries in the field. And so Sid was presenting his work on clonal hematopoiesis. I was presenting my work related to, germline genetic variants in exomes and loss of function mutations. And, we had a nice story related to ApoC3 that paralleled PCSK9, and that actually is now an approved medicine. Yeah. So I presented that work. Everyone there was engaged and they understood it. Sid presented his work, and I remember sitting there because, he came after me, and everyone was engaged. But when Sid talked about cardiovascular disease, everyone was like, we don't believe it. You've got a great biomarker for age. We know that age is a dominant risk factor related to cardiovascular disease. You just have a marker for age. So the output of that meeting was, Eric Lander said, you guys have to work this out. Is this just, correlative to cardiovascular disease, or does it actually have a role in cardiovascular disease? And it was pretty daunting for me because I'd spent, my postdoc working on, germline genetic variants, and I've been now asked to enter the field of cancer genetics. And Sid was a wonderful partner. Ben has been also a wonderful collaborator / mentor over the years. And we had lunch after that meeting, kind of strategizing what the experiments would be. What are the data sets? Again, even with human data sets, we would still need some, controlled experiments. So we brought in Peter Libby, who's a collaborator, former chief of cardiology at the Brigham, also, local, to do the mouse experiments as well. And that led to this, 2017 paper where we showed that, among individuals with coronary artery disease, including early onset myocardial infarction compared to controls, there was a relative enrichment for clonal hematopoiesis, and then modeling that in mice also demonstrated atherosclerosis. And that kind of set off a whole new area. And, as I said, that was tail end of my postdoc. my mentor say catheterization that was sufficiently different from what he was doing, so it allowed like a nice, exciting angle for me.
Yes.
And one of the areas that I took on.
And
So
at the time,
9-10 years later.
Yeah, and I mean the field is ripping not just in clonal hematopoiesis, but somatic mosaicism across every organ system. Like it's been systematically underestimated, I think, how important it is. And I suppose from a mechanistic perspective, is it really about inflammation? And what else is the story? And what can you say about what we know about how causal it is? And we'll talk about clearly, I think you think, and much of the field thinks it's causal because you're going after it therapeutically, but would love to, if you could talk a little bit more about what the downstream cascade is behind these bad clones that cause all kinds of problems.
Yeah, clonal hematopoiesis itself is a phenotype that relates to multiple different genes and driver mutations in those genes that converge on a phenotype of clonal hematopoiesis. Its disease-related implications are not exactly the same across these. And we've only started to understand that as the data sets have gotten large enough that we can look at, individual genes, sometimes individual mutations. So the top gene that is observed for clonal hematopoiesis, driver mutations at DNMT3A, their prognostic relevance to cardiovascular disease is largely no. I would say, unresolved in the field. If it truly does have an impact on cardiovascular disease, it's very, very small. But the number 2 gene, TET2, that's where there seems to be a clearest, consistent signal across most data sets. Okay, and it's interesting because both of them are natively epigenetic regulators, but they have opposing epigenetic consequences, but that converges on that phenotype. But there's some others that are not as commonly observed, like JAK2, V617F, P53, some splice site mutations. Identifying what their actual effect estimates still is hard because they're uncommon, so it's mainly a power issue, but generally appear to also have similar adverse prognostic relevance. But TET2 is where the signal is clearest. Now, the biology has converged along the lines of known biology of inflammation and cardiovascular disease. Unclear if that is the only one, but that at least is the clearest and the most compelling so far for a therapeutic hypothesis. So TET2 mutations in general, obviously clonal hematopoiesis, strong association with cardiovascular disease, modeling TET2 loss of function in bone marrow cells in mice in an atherogenic model also promote atherosclerosis. That was in our paper in 2017. And I should also give credit to Jose Fuster and Ken Walsh, who are across town at BU, who are also working on this and had a science paper on the mirroring aspects of this. Huh. And Jose has now been a great collaborator. Sorry.
Yeah, and just from my understanding is that what we think is happening in the TET2 clones is methylation is getting out of control, and that is resulting in the inflammation phenotype. Is that the simple version?
We think so, right? Like the inflammatory cascade that is most well established or well described for atherosclerosis is via the NLRP3 inflammasome. And to some extent, there is some relationship with cholesterol crystals within atherosclerosis, but that kicks off a separate inflammatory cascade that is activating IL-1 beta and IL-6. High sensitivity C-reactive protein, which is a clinically used biomarker for inflammation and cardiovascular disease, is largely a non-causal readout, but reliable biomarker for cardiovascular disease risk. And the therapeutic hypothesis has been validated for that cascade. There is a medicine called canakinumab, an IL-1 beta monoclonal antibody that's made by Novartis. They had a clinical trial now almost about 10 years ago, led by my colleague across town, Paul Ritger, showing that in patients with cardiovascular disease, elevated high sensitivity reactive protein, patients who were given canakinumab versus placebo had a lower cardiovascular disease event rate by about 15%. Okay. So as it relates to TET2, and this is work, again, we did, but also Jose, I'd say Jose took it a step further, really suggesting the causality and relevance of this pathway is that in the mice that are modeled as having TET2 loss of function mutations, a greater atherosclerosis, they also gave an NLRP3 inflammasome inhibitor. And they showed that those mice who didn't have TET2 mutations had a slight reduction in atherosclerosis, but the mice who had TET2 mutations had an even greater reduction of atherosclerosis. suggesting that there may be particular relevance in that pathway.
Yep.
Now, Novartis, because they were also, reading the literature, they went back to this trial called the CANTOS trial, and they did targeted sequencing of TET2 mutations in the patients in the CANTOS trial. And they observed that the patients who had TET2 mutations, they had a substantial lowering of cardiovascular disease events. So about 15% for the overall trial population. For the small fraction that had TET2 mutations, it was about 60 to 70% relative risk reduction. And so these are, exploratory analyses within, these clinical trials, non-pre-specified, but actually quite consistent with what Ken and Jose had observed in the mice. We said, okay, let's use our skill set in germline genetic variants. And it just so happens that there is a mutation in the IL-6 receptor that is quite common, a missense mutation. And about 40% of individuals have it. It's almost 50-50. And well described in the literature, papered out, almost 15 years ago, that individuals that have this common genetic variant have about a 5% reduction in cardiovascular disease risk. Statistically significant, but modest, but it's actually the best genetic validation of that pathway. Right. So we said, if that is critical in the pathway of clonal hematopoiesis associated with cardiovascular disease, if those individuals had defective signaling by having this common missense variant, are they going to have a greater relative risk reduction? And indeed, it was about 40% lower compared to 5% for those who didn't have CHIP compared to those who had CHIP and then also happened to have the IL-6 receptor mutation. So 3 lines of evidence that are converging strongly on this well-described pathway. And, actually now Jose and I were close collaborators. We did a similar kind of interrogation for colchicine. Colchicine is actually an FDA-approved medicine, an anti-inflammatory. That's a gout medicine and used for other inflammatory conditions. In clinical trials, been shown to lower cardiovascular disease risk. Nobody actually knows exactly how colchicine works, but we think it's through the NLRP3 inflammasome cascade. And similarly, in murine modeling, and then also looking at individuals who are prescribed colchicine, there seems to also be greater benefit for colchicine. And that's helpful to know because that's actually a tool that we have available to us today. Colchicine has some toxicities, but there's another validation of the relevance of this pathway. And there's now a lot of drug development in this pathway.
And maybe that's a good segue into TenSixteen Bio. You co-founded a company that is going after this therapeutically. I would love if you could, and my understanding is you're not treating the downstream inflammation, but you're trying to really go at the clones in a relatively targeted way. I'd love to hear a little bit about the strategy there. And I don't have a sense for what the patient population would be. Like, obviously you need to screen and identify who has clones, but what proportion of people, and what does that risk ramp look like? Like how would a trialist or a doctor look at a chip risk in context of all the other factors that influence risk and think about who would be a good candidate for that kind of targeted therapy?
Yeah, no, great question. So, just taking a step back, it's very exciting that there is a potentially new precision medicine paradigm for coronary artery disease. It's like us trying to become oncologists. In general, what we do today is we say estimated risk is high. And so if estimated risk is high, we'll hit kind of the pathways that are generally canonical for atherosclerosis. Here we say risk is high related to this specific pathway, and patients might benefit particularly more with this targeted intervention. So that's pretty cool. That's pretty exciting.
Yeah.
And so TenSixteen Bio, VC-backed startup, I had the pleasure of being involved in its genesis. But I would say that there are multiple different entities, including large bio and pharma, that are interested in this as well, too. Because ultimately, in deriving value, it's, really trying to identify the patients who are most likely going to benefit from the intervention, and then identifying them and then giving them giving those patients the medicines. Now, there are several challenges in drug development related to clonal hematopoiesis. It's not like a standard assay that we would use for atherosclerosis, like lipids, even high sensitivity C-reactive protein. That as an assay is widely available in clinical practice. It's not necessarily widely used, but it's widely available. And Lp(a), as you mentioned, is widely available in clinical practice. Now, targeted sequencing for CHIP mutations isn't necessarily that high, but most of these tests of doing just blood-based sequencing, blood-based biopsies, in general are high-cost assays, and they're not routinely done in clinical practice. There are lots of biobanks that exist. We have a biobank at Mass General Brigham. Other health systems do as well. Most of them have blood-based exome sequencing, so you can identify individuals with clonal hematopoiesis. But in starting new therapeutic programs, especially assessing for toxicities in phase 1 studies, ideally the patients don't have additional comorbidities beyond the feature of interest. Now, this is a particular challenge beyond finding individuals with clonal hematopoiesis. It turns out that the health implications are larger than oncologic and cardiovascular disease. We've described in collaboration with Ben Ebert's group relationships with liver disease. They've been involved in studies of chronic kidney disease. And all of these have particular relevance to new medicines that you would give these patients. So finding the clean individual that just has a CHIP mutation, maybe is on the path to atherosclerotic cardiovascular disease, but doesn't have liver disease, doesn't have kidney disease, turns out actually to be quite challenging. And that has been one of the near-term challenges when just kind of conveniently using patients that are in biobank, because usually time has gone on that they have developed comorbidities. Strategies that we have started to adopt, and one of my former trainees who is now well established at Vanderbilt, Alex Bick, has demonstrated this beautifully. If you go to the patients in the cath lab, the cardiac cath lab, that are coming in for fresh cardiovascular disease events, a lot of them obviously are enriched for having clonal hematopoiesis. But many of them don't yet have, the other comorbidities that would preclude them from other studies. So that actually has been a good source of patients to potentially enroll in clinical trials. We've started that also at Mass General Brigham as well, too. Because there's I was alluding to this, a lot of drug development to anti-inflammatories for cardiovascular disease along this pathway, many companies are also strategizing potentially drug development pathways for clonal hematopoiesis as well. But we're working on surmounting these critical challenges to actually, feasibly achieving that.
Yeah.
And I will say, it's like, I said 10% have clonal hematopoiesis when you get above, the age of 70. It's less common when they're younger. And then we're looking at not all individuals with clonal hematopoiesis, really focusing on TET2, which is an even smaller group of individuals.
Yeah. What does that percentage look like? And is the age ramp any different? Or I'm sure it is subtly different, but what does the TET2 subgroup look like?
Yeah, I mean, compared to it's about 20%, maybe 25% of individuals that have clonal hematopoiesis. Yep. And it's a kind of exponential as far as prevalence of clonal hematopoiesis. But that does bring up actually a really interesting point. So if you look at individuals that have survived to their 80s, 90s, 100s, many of them, sometimes even most of them, have massive CHIP clones. And so it leads us to ask, okay, is there a reason that clonal hematopoiesis exists? Some people believe, and it's not unreasonable hypothesis, that it is there to exist for when there is bone marrow failure later in life, that there are these clones that exist that kind of can take off and support hematopoiesis. And so what that means is that if you are inhibiting clonal hematopoiesis, might that actually lead to problems downstream when clonal hematopoiesis is needed? So disentangling what might be malignant as far as health outcomes from clonal hematopoiesis versus when it might be needed is a new separate challenge to uncover. I think what also supports this hypothesis is actually if you deeply sequenced individuals in middle age, I gave you those prevalences. So deeply sequenced is like 10,000-fold coverage. Almost everybody has a CHIP mutation, has a cancer-causing mutation that is quiescent, that is not led to clonal proliferation. So the questions are not necessarily of mutagenesis. What ultimately kicks off clonal hematopoiesis itself? It does support that these things kind of exist and are waiting for when they might be needed. So that risk stratification is needed and required. And now, there are a lot of ongoing longitudinal studies to help us map that. This is feasible because it's blood. You had mentioned about somatic mosaicism and other tissues. I totally agree. This is a tip off the iceberg, and the field started because of convenience, because there was a lot of blood available. It's easier to do. But somatic mosaicism occurs across all tissues. And that has a whole host of other outstanding questions, but that's very, very early in its field. I mean, we're involved in an NIH consortium coming from the Common Fund called SMOT, somatic mosaicism across human tissues, that is intended to really define, at a small scale to start, what is normal for somatic mosaicism. So, but yeah, there are a lot of complexities here, even with CHIP as a slightly mature field in the mosaicism space.
As a comment for the listeners, but also for you, tomorrow, as we're recording this, tomorrow's episode that's coming out is with Dr. Chris Walsh from Boston Children's Hospital, and we talked all about brain somatic mosaicism in neurons, and he described a very similar phenomenon. Actually, if you look in supercentenarians, you see plenty of mosaicism. It's not like they are, they're the lucky ones that escape it. It's there's clearly something. There's some inescapable mechanism here that is either it's important or it's inescapable, and selection is just not working hard enough on it.
That's right. And this is why you need the longitudinal studies, and Chris's work is really, his field is relatively mature because of his vision and his strategy and being able to procure neurological specimens across different disease states and then modeling it. I mean, you'll hear all about it from Chris, but he's been, wonderful collaborator and somebody that has been an inspiration for the work that I do. Yeah,
it blew my mind. I mean, all of this stuff is it does feel like we're rewriting our understanding of almost every organ system through, but often the brain is the last frontier because it's so hard. But they've managed to really get ahead of the curve on this. I was amazed. Yeah. What you were saying about the multi-system morbidity of this also had me, and your comparison to oncology had me thinking about, is there a basket trial type approach where you're looking not at preventing or fixing a particular phenotype, but you're looking at people that have this particular clone type? Because you've also associated different drivers with stroke and with other phenotypes, clinical outcomes, right? So there may be a way to think about this as genotype or clone driven in a multi-system way.
Yeah, and that's the strategy of going directly at the clones that maybe we have the crystal ball and can know that they're toxic clones, that you have the opportunity from a value perspective to prevent multiple health consequences for an individual patient. So that's the ideal. The inflammation is almost like, a Band-Aid, but if you can kind of do it earlier, there's a prospect of substantially more value. That risk stratification part is, I think, ultimately critical in just thinking about preventing long-term issues that might occur with preventing clonal hematopoiesis.
Yeah, maybe a good segue into talking a little bit about polygenic risk scores, because it seemed, at least by my reading of your publication record, you were working on these two paths and had an amazing, probably PhD postdoc and then first independent role working on both of these fields, and they're obviously complementary from a data set perspective, but really they played out in really different ways, right? Could you talk a little bit about? A lot of people listening to this will have a really good understanding of polygenic risk scores. You don't necessarily need to explain it, right. But what's your perspective? Two questions. One is, how independent is that from the CHIP signal? And then what do you see as the clinical opportunities for polygenic risk scores today? Yeah. I mean, polygenic risk scores came out of just my major focus on gene discovery and understanding mechanisms of disease. So this is of the flavor of germline genetic associations. The CHIP mutations occur in a small fraction of individuals, but I remind everybody is that for every trait, every health consequence, germline genetics matters. We are all made up of about 20,000 genes. Those 20,000 genes are necessary to manifest us. Not surprising that they would be important to manifest disease. And variation in how those genes are expressed, to some extent, explains the variability in the traits and health conditions that everybody has. And it's a balance at the population level for a trait, and then for a given individual, also a balance of whether it's genetics and not genetics. So as the field continues to mature on genetic discovery, meaning the data sets get larger, there have been more and more individuals, the opportunity to use the totality for that of information beyond biology for just overall risk prediction
has also matured. That's also been a reason for why I put more and more emphasis on this area of investigation as well. Because it's, what are the problems that it's addressing, right? So I'm, clinical preventive cardiologist, and I oversee a program in preventive cardiology. What that means, the patients that I see in the office, often younger individuals, often 30s to 50s, and they're coming to see me because the family history is the thing that creates some unease with them, with their primary care providers, that doesn't actually cleanly fit in any prevention paradigm today. The prevention paradigms today are largely centered on 10-year risk estimation, unless you have very abnormal lipids, which is, a very small fraction of individuals, about a few percentage. Cardiovascular disease is very, very common. So 10-year risk estimation. So 10-year risk estimation really works if you've made it to middle age and have not had any cardiovascular disease events. For many of those individuals, there could already be an accumulation of atherogenic signals and atherogenic consequences. And then we're just, trying to prevent what we can estimate as high-risk disease in the 10 years. And we've been using that heuristic, since the first inception of prevention guidelines. That's the 1980s. And that served us very well. But cardiovascular disease continues to have that dubious distinction of being the leading killer in the US in the last couple decades across the world as well, too. So what we're missing is the signals earlier in life. And, the one thing that we have today is if they have severe hypercholesterolemia that's greater than 190, that's, about 3 to 5% of individuals. So not insignificant, but among the reason that also since the 1980s, we say that people need to get screening lipids in their 20s. So we do that. But the question is, can we identify many more? Now, the genetics is knowable information earlier in life. And the risk factors that estimate 10-year risk, in general, they're starting to manifest in middle age. They do manifest in younger individuals, but these 10-year risk estimators are largely a function of age and sex. The individual risk factors will move the needle a little bit. You could actually entirely remove them, and you'll get most of the exact same score just with age and sex. That is the inherent limitation. That's a known limitation. So my patients that I'm seeing in the 30s to 50s, they'll have a family history, but like that, it's almost impossible to be high 10-year risk when you're at that age, just given just how they're put together. Okay, so polygenic risk score, which is just, basically a weighted summation of each of the risk alleles that are associated with coronary artery disease cases relative to controls. The methods are evolving, but largely what determines their overall performance have been the data sets and the number of different individuals that they're seeing. So each of these genotypes in different genetic backgrounds will better refine the individual signals. And the performance of the scores as far as odds, prognosis, continues to increase. Now, interestingly, like the model fit, so the adjusted R squared or the C statistic, which is kind of the discrimination of cases relative to controls, all of that is actually relatively comparable to each of the traditional risk factors. I say they all kind of move the needle a little bit, but it's all actually about the same of everything that we're doing today.
Right.
But the prognosis continues to go up. So if you look at the individuals that have similar risk as severe hypercholesterolemia, where we say, okay, we've got to do something earlier in life, about 5%, that's about threefold estimated risk. It turns out for that same risk mode, about threefold risk, that's the top 20th percentile of contemporary polygenic risk scores. So 1 in 5 individuals. So that is a signal that is knowable also for individuals in their 20s, potentially earlier. There's a lot of complexities in thinking about earlier, but at minimum, in the 20s. And even just from like a simplistic patient perspective and the clinician perspective, we're always asked to interpret the family history. People do a lot of things based on the family history, but family history, self-reported family history is very complicated, right? So somebody might be young, and so their parents are young, so they may not have manifested disease. Or let's say an uncle developed a heart attack in their 40s, but was also a smoker. Was it all just related to smoking? Yeah. Or mom died early because of breast cancer and never had the opportunity to manifest cardiovascular disease. is very complicated at the individual level. This is actually about family history.
PhD in statistics to sort it out, right?
Well, yeah, and this is also why it's just not in these guideline-supported risk calculators, because it's very specific to that patient there.
Yeah.
And there are many different things that you share with your family. You share DNA, but you share a wide variety of health practices, education, income, beliefs of health. So this is a way to say, okay, is there a through line for the patient, for the person that is not in the room, that I can ask more questions or get more information? Is the genetics the relevant part? So that's the part that really I think the clinicians understand, that makes sense to me. It also makes sense when I'm talking to patients, is it can really help us refine those signals. So there's at least a nice, clear clinical construct. But even taking a step back and say, okay, what are we missing? Are those are the early risk signals. We have one with severe hypercholesterolemia. I think the next could be, polygenic risk scoring, and it has a very, very similar evidence basis based on the things that we're doing today. Lots of other practical challenges, and maybe we can get into those things. But I think there is a nice opportunity. So in the last guidelines, the last cholesterol guidelines, it was the first time any major clinical guidelines has adopted polygenic risk scoring. It's not strongly in there, but it's actually the first time. And if you look at like the timeline for things entering in, like Lp(a), which you mentioned, that has been a biomarker for for 20 plus years.
Yeah.
Only more recently has been introduced in guidelines and sort of increased in its enthusiasm and use. And this is the first. This is the start.
If we were to go 10 years in the future and we have time to continue working on the science and sort out all these bottlenecks, would you say the biggest impact of polygenic risk is going to be in being part of the screening apparatus, so we can detect people earlier? Is it going to really drive the way someone is treated, in the sense that you're gonna use that score to say this collapses on a particular pathway and so it's guiding treatment versus just risk overall? Yeah. Is it helping us understand the fundamental biology in some way that we're designing trials differently? Like, where do you see the biggest impact amongst those, or a fourth that I'm not smart enough to think of?
Yeah, no. I mean, it could be all of those. I think the risk is a framework that is used today in escalating therapy. When I think about how to get these into the clinic, engage clinicians, because I wear the two hats, speaking with scientists and then speaking with clinicians, is you have to think about what is the evidence basis of what people do today, why are they doing the things today, and you have to first harmonize with that. You can't immediately come in and disrupt, or at least think that you can immediately disrupt, because that will not permeate into practice. It will not permeate across clinicians, not across patients. So the risk-based framework is the way to get started, and there is a similar evidence base. Now, interestingly, because the risk is a means to giving a therapy and better identifying patients who are going to benefit from that therapy. And the only reason why we do that is because basically the relative risk for everybody is about the same when you benefit from cholesterol lowering. Statin is the primary preventive medicines that we're going to give. And just if you have higher absolute risk, relative risk reduction is the same, but the absolute risk reduction will magnify. So you're more likely to experience that benefit. But it's also practically very challenging in primary prevention, patients who haven't had an event. Because if they never go on to have an event, either your recommendation worked or it didn't work, and nobody wouldn't know. Like, so you just have to you have to make sure that you have conviction and the patient understands, and you're able to communicate that to patients. Now, interestingly, just with the overall risk predictive score, kind of putting everything together, if you go back and look at clinical trials for both statins and PCSK9 inhibitors, LDL cholesterol lowering medicines, the relative risk reduction for the same amount of LDL cholesterol lowering is greater for those who have high polygenic risk. I'll say we don't have actually a good reason for why that occurs. It just is a recurrently observed observation across subgroup analyses and clinical trials. So not only is the absolute risk higher, relative risk reduction is greater. So absolute risk reduction is further magnified, meaning you're even better with this tool at prioritizing individuals who are going to benefit. Because again, the risk prediction is a means to identifying the patients who are going to derive benefit, or better identify patients with benefit. So this has unique properties for that aspect. It kind of relates to the first 2 points that you said of, maybe treating patients differently. This is still vanilla statin lowering.
Yes.
Whether this can be done for other therapies, hard to know, but it might be a mix of other polygenic risk scores. We're doing some work in this. Others are doing some work in this. Again, I'm really interested in better identifying what are the pathways that are highly relevant for the patients, and then identifying the therapies specific to that patient so they're more likely to benefit from that as well. There is, I think, a nice opportunity for clinical trials, because for cardiovascular disease today, most clinical trials largely occur in patients who already have cardiovascular disease.
Right.
There are many different complicated reasons why. A lot of it is efficiency, because those are the highest risk patients to have an event if they've kind of already had it once before. It turns out, actually, if you look at all the clinical risk factors that we look at today, and many of them are well managed across these patients. So what is the through line for why patients get recurrent events? Turns out among the top is actually a polygenic risk score. So as already standard practice today to improve the efficiency of clinical trials is take those who have cardiovascular disease and some other risk factors, because those risk factors are a proxy for higher risk for recurrent events. If you added in a polygenic risk score, that would increase the efficiency of your trial as well, too. So I think there is an opportunity for that.
Does that question on that, when you're designing a trial and considering that trial design strategy, let's say you enriched with the top decile PRS carriers, how does that impact the interpretability of the results and or the label down the line? Like, it's a very naive question, but does it reduce do you have to think about that when you go to the pivotal trial of,
yes.
Does this generalize?
Yes. So this is probably the most important problem right now. And this is kind of what I was alluding to at some of the practical challenges. So if you go to, Mass General Brigham or UCLA or Stanford and get lipids tests or lipoprotein little a test or HSCRP, you'll largely get the exact same result, within variation if it's, not the exact same blood sample. So largely the same. And so, you would have confidence in any of those. Now, because there is not the single canonical polygenic risk score for anything, and there are different ways to calculate, if you go to each of those different entities, not everybody has polygenic risk scoring, but there's different companies. You don't know exactly what
they do. Doing. They're not the same.
You won't get the exact same result. And there's actually a really nice paper describing this from Penn, and we had one as well, too. So this is a practical challenge, just even today in ordering polygenic risk scoring, but also in the clinical trial design. So which polygenic risk score will you use? Yeah. A lot of this is a function that the field is continually evolving. Our ability to interpret the genome and quantify in this manner is also evolving as well. So if you initiate a clinical trial with a polygenic risk score, in 5 years for a predictive polygenic risk score, there is going to be a new one and better. So there are different ways to do this. You could lock in that specific polygenic risk score, and that's fine. I know many folks are thinking about that. I'll say the FDA from a pharmacogenomic perspective is largely still thinking about single SNPs, but this might be a little bit different when it's like a label. It's not necessarily a pharmacogenomic.
Yeah, it's not like
through a standard manner, standard interpretation. Yeah. The other is looking at kind of how things are being handled with artificial intelligence. The FDA has kind of a pre-specified computation change that is allowed, a pre-specified control change plan that you can use for AI if the algorithm changes within your development time, and then you have pre-specified heuristics on what you would change and how you would change. So you could do something like that and then kind of go back and reanalyze the trial, because it's just, mathematical calculation for a new polygenic risk score. That's the part that I'm excited about, because it kind of parallels what other things are doing with the artificial intelligence. Obviously, much more complexities. These are largely logistic regressions in how these scores are kind of coming together. So I think that's a viable path, but it's an untested path so far. So that's the clearest limitation right now to drug development, but that's a surmountable challenge.
Yeah, that's it's a big one, and probably one you're going to be thinking about a lot going forward. You're on the cusp of a really big move. As we wrap up here, I'd love to just talk a little bit about I think in a couple of weeks, you're moving to Amgen. Yeah. You're going to lead cardiometabolic and human genetics research. I'd love to just hear a little bit of the story there. What made you decide? It's probably the original genetics-driven drug discovery pharma in a way, right? They bought Decode back in 2012. It's always been part of the DNA of the company. Yeah, tell me a little bit about what made you decide to make that move.
Yeah. I mean, I guess depending on when the listener is listening, I might already be at Amgen.
Yes, that's right.
So yeah, August 3rd is when I transitioned. Very excited. Yeah, the opportunity to lead, two different functions within Amgen, super exciting. And part of it is, major part of it was the cultural alignment. It's a company that has valued human genetics in the development of medicines. And as a result, they have taken big, important early bets that have paid off. They were the first, I would say, along with Regeneron and Sanofi, to go after PCSK9 as a target, and did it quickly, and are now the most successful with Repatha or evolocumab, a medicine that I prescribe all the time to patients today. So to oversee that aspect of it is fantastic. And one of the early folks on Lp(a), and they have a medicine on olpasiran. And their obesity medicine, Maritide, which is a GLP-1 receptor agonist, also a GIP receptor antagonist. Yeah. That's actually quite different than tirzepatide from Lilly, which is a GLP-1 receptor agonist and a GIP receptor agonist.
And that comes from the Decode. Does that come from the Decode?
Exactly,
work, right?
From analyses in Decode that showed that loss of function mutations in GIP receptor are what lead to lower BMI, and that led to both. It is interesting that empirically, obviously, we know that tirzepatide leads to lower increased weight loss compared to GLP-1 receptor agonism. And there's some data that suggests that if you agonize to that degree, you're basically creating functional antagonism, but this would be kind of clear pharmacology. Anyway, all of those together suggest that human genetics has been very important for Amgen, has paid off for Amgen as well too. And I know, many of the senior folks at Amgen, actually the, people who helped make this happen were Jay Bradner and Howard Chang. Jay is the head of R&D at Amgen, was previously the head of NIBR, with Novartis Institute for Biomedical Research. But before that was a physician scientist oncologist at the Dana-Farber. And I had the privilege of working with Jay as my attending when I was in residency, actually in the first year when I was an intern as well. So I maintained a close relationship with Jay over the years, and he'd gotten to know me and my work and really thought about, the opportunity that would make sense for me, but also the need that they had at Amgen and what they were thinking for their strategic vision. And Howard Chang, who had just came on from Stanford in HHMI last year to be CSO, and work closely with Jay. Somebody like Howard, who is, one of the inventors of ATAC-seq and really a visionary in RNA biology, and understanding how he strategizes his science overall and thinking about that overall impact and what led him to transition from academia to industry really spoke to me in thinking about impact. And now having more tools, significant infrastructure, and working with people that I respect and collaborate already from the academia side, with industry, very, very exciting. So, I'm really excited for this next step. Hopefully, I can bring many of these themes forward, but expand into many new areas along the field. And then the last part, with Amgen being in the West Coast and really embedded in the heart of artificial intelligence and drug development, and seeing and understanding how they've been doing it in a responsible way, in a strategic way, that also really makes me excited for, where things are going.
Yeah, absolutely. Well, it's exciting. They're very lucky to have you, and it sounds like an amazing match. Is there anything you want to cover before we close out? Otherwise, I just want to say thank you for sharing all this amazing research with us, and you've given me a ton to think about. I know the listeners will appreciate.
Yeah, no. I think we covered a lot, and I'm always available for any follow-ups. I'm really excited to hear about what you talked to Chris Walsh about as well, because I'm sure I'm going to learn a lot from that too.
Yeah, it'll be out tomorrow. I'm actually I scheduled in with Sek Katharason in about a month as well.
Oh, so wonderful! Great.
When we stop record, I'll ask you for hardball question that I can throw his way. That only you were a student, right, or postdoc?
I was a postdoc in Six Club. Yeah.
Yes. So no, I'm really looking forward to that. So yeah, it's one of the best hours of my week to speak to people like you. So thank you.
Thanks, Patrick.
Thanks, everybody. As always, we'll speak to you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.